For lab this week, we've got some examples of plants that are flowering and fruiting from our greenhouse. And I'll walk you through them and maybe a little bit about what to pay attention to as we're noticing them, and maybe trying to identify features about their respective reproductive parts. So I've got them kind of loosely organized. There's some that are closely related to each other at the middle table here. This group is all closely related to each other. And then there's a group at the third table over that are also pretty closely related to each other. So if you were in person, you'd get a chance to dissect a few of those. Might be interesting to compare the differences and similarities among things that are closely related and kind of contrast those with plants that are a little more distantly related. So I think I'll probably just walk you through what we've got, focusing on the reproductive parts and some material that we've learned about already. The first one here, this is a plant called Peperomia. Sounds a little bit like pepperoni. It's related to black pepper, and it's got these weird things coming out, and they're so small. What in the world is that? This is something you'd want to get under a microscope to take a better look at, but basically these are reproductive units, and they would count. Let's get a little closer look. I think probably after the fact I'll come around and try to get you some microscope images of these, but this is a flowering stalk. I can do, as we're learning about these, probably a good idea to do some sections. So I can do a longitudinal section through this axis. That doesn't tell me a whole lot except that it's solid inside. So the reproductive organs we're looking for might be smaller than this. Cross section wouldn't tell us a whole lot more. So like I said, you basically have to get this under high power magnification, but those little dots on there, each one of those is a flower. And they're highly reduced in this group, and they would have the reproductive parts, but we've got to get them under the microscope to see that. But we can put a name on how their flowers are arranged. So we have a growing axis here of reproductive tissue. Little dots along it are flowers. Those flowers are so small, they certainly don't extend from the axis, which would make this a unbranched, right? Not branching here from the point of origin. And the flowers are not on stalks. So that qualifies this as a, maybe I won't give away the answer. Maybe I will. This one counts as a spike, right? So you can see the reproducing units here along the plant. So this is another spike where it kind of goes from a piece of stem without any flowers to a piece of stem that now has flowers. Some of these are just getting developed, so you can kind of see different ages there. There's a bract. So this is a leaf associated with that reproductive axis. Here's another bract. Sorry, there's a bract with an inflorescence in it. So we could peel this away and verify what it is. And I don't know if that's going to come across too well on the video, but that is a sheathing leaf. So a leaf associated with the base of the inflorescence, that makes it a bract. And this little part here, that would be stalk right under the flowering part. Leading to the flowering part, that would count as our peduncle. And individual flowers, I'll have to show you a microscope image of those because they're just not going to come through on my camera here. All right, let's move over to the middle table. I chose some that I thought might be open by tomorrow. This is the end of the day before the lab happens tomorrow. So we can see that we got some flowers not yet open. There was one, where did it go? Here's one. This one is just a little bit showing like it had some open parts. Maybe we'll sacrifice that one. Okay, so we've got, unfortunately, lots of small things here. Again, probably end up getting these under the microscope to show you better. This is a good lab to do in person if you can. But we can cut longitudinal section is a good way to find out more about what you're looking at. I can see the bright yellow color. That's standing out to me. And then, of course, there'd be something wrapping it up. So bright yellow is probably our pollen color, which would indicate where the anthers are. Right now it looks like that length there is anther. I see we're not getting in focus all the time. So I'll try to get you a microscope image of these flowers as well. As far as the inflorescence type, those narrow categories of raceme, spike, and panicle, they go so far, and then they aren't always 100% descriptive. So this is an interesting one where it mostly looks like a raceme. The flowers are on short stalks, but the flowers also come off, looks like 2 at a time on this little side axis. So that would technically be some branching in the inflorescence, which in those narrow categories would qualify this as a panicle. So you might describe this as a narrow panicle, or always useful to use more descriptive terms than just slapping a label on there and assuming people know exactly what you're talking about. Here's another one. This is a good example of a acaulescent plant. So we've got a bulb down at the base. Bulbs, like we talked about in vegetative morphology, leaves basically around the base, and then a portion here that is the scape, leading to, again, flowers that aren't really open today. I might have to leave those be, because I think we'll get one open by tomorrow. But you can see, I think, looks like they are not branching, so the overall structure is pretty narrow here, close to the main axis. Each flower has a stalk on it, so that would be unbranched. Flowers with stalks, that would make that a raceme inflorescence. Here's another case of an inflorescence. I was going to say raceme, but then I have to check here for flower stalks. Really, it's a matter of any amount of stalk qualifies as a stalk. So that's a little bit of stalk there. That's going to count as a pedicel on this flower. The thing that's falling apart is the bract. Often there's one bract associated with each flower, sometimes a little more showy than other times. So a little stalk leading to the flower. And this one is maybe big enough that we could make it work for us on this little camera. Let's count our parts. 1, 2, 3, 4, 5, 6. So this falls into the monocots group, as we will learn in a little bit, with 6 perianth parts. It is bilaterally symmetrical, so the shape is not quite symmetrical in all directions. So you can see there's a bit of a top-down orientation here. But the color and the size of the perianth parts here, not all that different. So one thing we probably would use as a term for this, you might call these tepals, right? So we see them as they've got green streak down the middle, white on the edges. There are ones that are more inner than others. So sometimes they're referred to as inner tepals and outer tepals. Sometimes that stands in for petals and sepals. So instead of really calling these petals and sepals, since they're not all that different, and they all appear to be contributing to the same kind of function here, I think they would more properly be called tepals. So inner tepals, outer tepals, 6 of them total for a good monocot number, kind of in the lily family in the broad sense. Here's a question. How about fusion? So you've got tepals that are pressed together along the length there. Are they fused? Well, let's try. So notice I'm peeling this away, and it is in fact letting go, right? I didn't have to tear anything to get there. So that's a unit all unto itself, and it was not fused to others of the same kind. He loves me. She loves me not. No. Okay, so I could keep peeling those off. That might be a good way to get in there. I think it's kind of nice also to see the attachment of organs. So I'm going to leave that a little bit intact and do a longitudinal section. So for most things, the longitudinal section will serve you best. I'm going to come up from the bottom, actually, on this one. If you chop down from the top, you might risk displacing a whole bunch of stuff. Got some pretty sharp shaving razors here. And I'm going to do my best not to cut myself on an educational video. All right. Kind of using my naked eye off to the side, I still can't see too well, not as well as I would with some kind of magnification. But again, we've got yellow, that pretty standard color for pollen, or at least the most common. So anther on top of a filament, looks like it's unfused. There's a fatter green bit right in there in the middle. It's kind of elliptical shape. Looks like an ovary, so that would be above the point where the other organs are attaching. Superior ovary, nothing has shown itself to be fused yet, so unfused tepals, stamens not fused to anything. And then maybe under the microscope we could see more about the gynecium. I can't really tell anything about it except where it is and the fact that it's there. So we have a hermaphroditic plant with bisexual flowers. And more to be told by the time we get this under magnification. So that was a pretty good one. That was not a large flower. That was like 1 centimeter long. Still one of our longest, biggest ones to work with so far. Here's a nice, refreshing change. This is a plant with some very large flowers, very obvious organs on there. And I'm excited to get in and check this one out. This is a Christmas cactus. Genus has one of my favorite genus names to pronounce, Schlumbergera. And I'm going to try to get, we'll go ahead and get one that's in good shape here. All right, I'm going to cut it, and we'll go ahead and take some stem under it. So there are these little stem units. This is in the cactus family, and so these are flattened photosynthetic pieces of stem. There are no true leaves in this plant. And so this right here is the end of stem, and then a transition, the end of one of those stem units, and then kind of flares out in a different color and a different shape, a little more three-dimensional than we saw in the others. And here we've got some, what do we got? Petals, tepals, I can't tell. Well, they all kind of look the same. So we'll go at the base here. The first ones coming out are red. The first one here was really small, but then they've just gotten bigger gradually. This gradual transition from things that started red and finished red, I think I'd probably safely call those tepals also. Just not enough differentiation. And we'll get to see in there if they are fused together. I think maybe we'll try to explore this before we take it apart too much. This is a group that has lots and lots of stamens. So just like a paintbrush worth of stamens up here. The white filaments, again, topped with anthers that are kind of lemon yellow. And then a different structure here in purple. This one is just like kind of a globular stigma. I don't know this plant in fine detail. Sometimes you have a stigma that is like closed up and then it opens later. So this one may open later. One way to answer the question of whether things happen later would be to investigate an older part of the plant. So here's an older flower that's long past flowering. We could check and see what its stigma looks like, and I can't even, okay, I can kind of find it. This one might be a little too far gone, so that's sort of where the stigma used to be, that purple line there. Let me try just a couple more. Here's another one that's on its way out. So this is a plant that has pretty continuous flower availability. So that flower, even though its flower is fading, there's not much. I don't see the stigma opening into any shape. Although if I kind of beat it up, it splits out into little lobes there. So I don't know if it did open and then closed again. That's also possible. There's a lot of intricacies to flowers that we don't really get to observe if we're only watching them at one point in time. Nice illustration here also of what happens to an older flower. So these are all the showy parts. This is the perianth parts and the stamens. Most of what's here is not really needed as this goes to fruit, and so it's fading, it's wilting, it's gotten kind of sticky. It's just kind of giving up and letting most of that flower tissue fall apart. All right, let's go back to our original here so we can see we've got Lots of stamens. Looks like a fused pistil of more than one carpel. And I'm going to try to do a nice longitudinal section again. We've got one here. Again, this is bilaterally symmetrical, so all of the stamens are off to a side. It's oriented now how a pollinator might approach it. And this one has a good look of like a hummingbird pollination. So animal coming to approach it, stick a narrow beak down there for some nectar at the base, and in the process contact these reproductive pollen distributing and collecting organs for the purpose of pollination. So when you get one of these, imagine you're a hummingbird. All right, let's see what we get with a longitudinal section. A long flower to get through here. Okay, lots of some juice down at the bottom. Is probably some nectar being produced there. Got a pretty good slice there. I didn't cut the style, but you can definitely see here where the style is differentiated from the filaments of the anthers. All of these are long so that they can reach out to the end of this flower tube, but they're connected, they're attached at the base, and the female part, the pistil here, actually has to attach to the base where the ovary is. And the green part was our stem, and then this more white part is the ovary. And so the ovary, we can see, you can't really see ovules in there, but I can just see kind of the pattern I would expect for things getting bigger, kind of a little bit of a grainy appearance there. And this is under the point where those organs are attached. So the style attaches at the top of that, as do the perianth parts and the stamens. So that makes this one an inferior ovary, which is something we see in the cactus family. Let's check those perianth parts. Are they fused? I'll just stick with calling them tepals. Are the tepals fused? So look at this. I got to tear that to get it apart. So right now I'm tearing tissue. Tearing some tissue here. And it looks to me like there's no way to get these off without tearing something. So the perianth was actually fused to itself. Was anything fused to it? Let's take our stamens apart. Oh, this is getting interesting. Ugh. All right. So we're also not able to get all of the stamens apart without tearing them. So they're attached to the perianth as well. So I guess technically we have a little bit of hypanthium. They're fused for a small portion. And then beyond that, the perianth is still fused, but the stamens are unfused from that moving ahead. So inferior ovary, perianth fused to itself, perianth also fused to stamens. Style is free from everybody after it leaves the ovary, but because everything is fused at the ovary, then you would also say that all these organs are fused together for at least some part of their length. So pretty exciting there. I'm glad we could share that one. I'm going to try just doing a cross-section through the half of ovary that we have left here. I don't want to destroy another lovely flower just yet. And as suspected, I can't really tell anything about placentation. Maybe if we get this under the microscope. So it's good to know about placentation, but if your flowers are small, you don't often see it. All right. Moving on. So we got a table here of plants that are related together. This is in a family called Crassulaceae. And maybe I'll just grab one from our kind of bigger member here. This is a plant called Chandelier plant. You might guess why. Actually, I think this one might be the one called alligator plant. There's a couple of related species. All right, so lots of flowers here. We can see there's some branching. We'll get in and talk about how this is a distinctive, different version of the panicle. One feature that it has is that all these flowers are basically born at the same height, something we don't see in kind of more generic panicle. So let me get to one of these, definitely a stalk on there. This plant's got an interesting kind of waxy, rubbery feel to it. Coming in from the outside, so unlike the cactus that we were looking at, this one has some sepals. See if we can count them. 1, 2, 3, 4, 5, I think. 1, 2, 3. Nope, I think we're at 4. Sorry. Kind of hard to count things in a circle, huh? So 1 on top, 2 on the sides, and then if I flip it over, there's 1 on the bottom. So I think that's a total of 4. 4 sepals. They are, see if they're fused to each other. So, yep, I've got to tear a little bit to get them apart. That's fused to other sepals. Looks like if I try to peel it away from the petals, though, I think I get all the way down to the base. So that's kind of interesting. Yeah, I was able to take that all the way apart from the petals without tearing anything. So sepals fused to each other, petals not fused to sepals, but petals fused to each other. So I would have to tear to get these apart. So this is a corolla tube. Let's look down at it before we start dissecting. You can see some stamens in there. 1, 2, 3, 4, 5, 6, 7, 8. Without knowing anything else, I would expect maybe, since we've got 4 petals fused together, we might expect a multiple of 4 for the number of stamens. So 8 seems reasonable there. We could definitely confirm that with other flowers or more careful counting. I'm going to do my standard longitudinal section here. Dun, dun, dun. All right, here's a good one. Okay, so I don't know if we can count parts since I threw away half of them, but I have isolated the other parts. So pulling this apart, the stamens are fused to the petals for a little ways. So there's a little bit of a cup there, but nobody's fused to the carpels, which are then part of a superior ovary. And actually, how many ovaries do we have? Well, I can peel these off separately. So this is a carpel, that's a carpel. It looks like we had a total of 4 carpels, I'm gonna say. So 4 separate carpels, 8 stamens fused to the corolla. Corolla tube is fused petals. And then not fused to that, but fused to itself was the calyx. And then this, because it's got separate carpels, would have what we call marginal placentation. And the fruit type would be one of those single carpel fruit types, and then an aggregate of those. So, no fruits on these, but the far table, I grabbed a few plants that are producing fruits. We saw this one last week, a parlor palm, and it's on the small side, so I don't know how well we'll get in there. But the overall structure, this is definitely branched, so on first blush, I'd call that a panicle. Individual flowers, looks like they are sessile flowers, and then developing into some kind of fruit. Not easy to tell on this scale. They're not done maturing either, so hard to say right now. Could be a berry, could be a nut, could be a drupe. Another one, this one's in the pineapple family. And this is a nice illustration of the transition from flowering to fruiting. So I happen to know, or I guess we can see some evidence. We'll grab this one. This was a plant that was flowering not too long ago. These are petal parts and long anthers, again yellow, and I can actually dust out some pollen on those. And then a cute little, I don't know if you can see it, spiral, like a corkscrew stigma on the top of the pistil there. All right, so petals, we have at least 3. I think I wouldn't be surprised by 3. I think 3 is a standard number for this group. Looks like they're separate for a ways. There's this here, Has the look of sepals, 1, 2, I think we're 3 of that kind of that organ also. And then this has a look of an inferior ovary. It's big, kind of beyond where things are colorful for the perianth. And maybe, let's find out. Back to sectioning. So this is also a nice big flower. All right. So I said it looked like an inferior ovary, and actually kind of tricky. You see there's some open space in there. I think that's part of our locule. So that's part of the open space inside the fruit. And then there's this horizontal line going across. That would be where the top of the ovary stops, and the other organs are fused to it. So kind of interesting. It's sort of a perpendicular line going straight across there. Let's see, perianth parts. So if we have an inferior ovary, then technically all the other flower organs are fused together because they're fused at this ovary portion. So sepals are fused together for that part, but then they're separate beyond the ovary. Petals, looks like they're separate beyond the ovary. This one's given me some nectar, so my fingertips are now very sticky. I'm probably going to be sticking all this flower stuff together. Stamens separate from each other after, above the point of the ovary, and then style separate at the top there. I think if we get the attendance we did last week, I probably won't have so many people that everybody needs a flower. But I'm going to be conservative for start and see if we can look at the ovary and cross section. I'm going to change my mind and go in for a full circle. Oh, beautiful. So first, full circle cross section through the ovary. I can see that I've got 3 locules. So that probably almost certainly tells us that we've got 3 carpels that make up the fused pistil here. And the ovules, they're just kind of little white grains in there, but they're, you see, emanating from the center. So they're coming out from the middle or the axis. So the central axis, that makes them axile. So axile placentation, 3 locules, and pistil made up of 3 fused carpels. So that was a lot of fun. All right, I got one plant left that has produced some fleshy fruits for us. This one, I don't remember what to expect on these. So they're going to be fleshy, either drupes or berries. And inflorescence type, you can see we've got some leaves. So some plain old vegetative leaves are associated here. That kind of confuses the terminology. So you could think of this whole thing as an inflorescence unit. Or you could think of it as units coming out from the axils of leaves. Which is, I think, how I'd rather define this one. So it's like an axillary, most of these, an axillary pair of flowers. Here's one. Whoa, forgot this thing was top heavy. Da-da-da, da-da. All right, here's one. So I saw a couple axillary pairs. Here's an axillary foursome. So coming out from the axil of this leaf is a little inflorescence branch. I'll go ahead and cut that off to illustrate the unit that I've got here. So 4 parts. Here's some evidence at the base. Oops, covering up the image. At the base of this fruit, we've got some evidence. Those are probably sepal parts remaining, and they're located toward the pedicel end. So that would make this a superior ovary. And this is sometimes, I think this may be where the origin of the idea of a stigma came from. It's just like this spot at the far side of a fruit. So that's where the style would have been attached, but that was in flower. Now we're in fruit, and there's a much different investment in the fruit phase. So I got a few of these. I can take a couple different slices. Fruit, probably, I don't know, you'll learn something from longitudinal and cross section. So I'm just going to go in with a cross section for starters. And I'm going to proceed gently because if I hit something hard, it's gonna possibly send my razor blade elsewhere. And I like my fingers. Oh, it's lovely. All right, what did we learn? There appears to be one seed on the inside. So there's something central, it's shiny. I think I just sliced through the seed. But I can squeeze this and determine that there's only one seed in there. And then maybe by plucking it out, okay. So I kind of got rid of the flesh. I'm left with a hard part. The question, oh, it is hard. The question of whether we have a drupe or a berry really comes down to whether the hard part of this is made out of seed tissue, which is possible, or endocarp tissue, which is possible. So if it's made out of endocarp, that would make this a drupe. If it's made out of seed coat, that would make it a berry. And I don't know if I'm ready to say just yet. Let me try one, just kind of mush it up. And if I can isolate a nice shiny seed from there, a lot of times that's good advice, good evidence that we've got just naked seeds. That looks like a seed. I think maybe this little circle at the base, I think that's an attachment point. So seeds have a little scar where they were attached to their mother. It's a belly button on our seed. And I think a seed. So I always, if you're really studying a group, you're probably familiar with what kind of fleshy fruits it makes. But I usually just double check against the description of the plants. So I'll probably do that and just confirm for myself that this is a berry. That was my first feeling is berry because it's smooth, pretty clean transition from fruit flesh into hardness of the seed. All right, so that's the plants we have in the room today. I might also supply you with some images. We'll see what I come up with.